Hello everyone, in this video I'll be explaining on the application or functions of those topic structure of bacteria and the application of bacterial growth. Coming to the structure of bacteria, the bacteria consists of various structures: the cell wall, cytoplasmic membrane, cytoplasm, capsule, flagella, fimbriae or pili, and spores. The cytoplasm contains all the cytoplasmic inclusions and also the deoxyribonucleic acid. Beside these components, in some bacteria, they produces a secretion on their surface covering the entire bacteria, and then if these secretions are in a loose form, then they are called as slime layer. But when these secretions are in a definite shape or structure, then they are called as capsule. And this capsule, it enhances the bacteria against virulence by inhibiting phagocytosis. Phagocytosis is a mechanism by which it engulfs the microorganism or the foreign particles and remove them from the body. Next is it acts as protective covering against antibacterial substances. So there are certain drugs which are given to in order to kill the bacteria, and this capsule will protect the bacteria against such antibacterial substances action which are directed against bacteria to kill the bacteria. Next is spores. Since these spores are highly resistant resting stage formed in unfavorable environmental conditions, so this structure of spores help the bacteria survive for a long period under unfavorable conditions. So even though the bacteria is being exposed to unfavorable environmental condition, it will survive that such harsh environment by transforming themselves or forming spores, which helps or survive the bacteria for a long period of time under unfavorable condition. Next is pili, which is also called as fimbriae, and this is a rigid structures and shorter in length as compared to the flagella. And this pili has a primary function to attach, help in the attachment of the bacterial cell to the surface of the host cell and helping in the attachment of those bacteria to cause infection. Coming to the bacterial growth, the bacteria are grown in the lab for the diagnosis or detection of the microorganism in the sample. So in order to grow in the bacteria artificially in the lab, it requires certain elements, essential elements, minerals and other organic growth factor. So the essential elements which are mainly required here are hydrogen, oxygen, carbon and nitrogen. In addition to this, phosphorus and sulfur are also required. So various conditions and nutrients should be kept in mind while growing the bacteria. And why is it necessary for growing the bacteria? It is to in order to diagnose or detect the causative microorganism that is present in the sample.